We're now going to talk about all of the structural reasons bacteria are not like us. Um, Bacteria themselves, as a class of microorganisms, have several structures that are shared by almost all of them. For example, a cell wall. Almost all bacteria have a cell wall. Um, they have a nucleoid. So because they're prokaryotic organisms, they don't have a nucleus, but they have a nucleoid. Their chromosomal structure and their replication of that genetic material is also somewhat shared across bacterial organisms. And these are all things that are different from how we do them. So we're going to highlight what is integral to the bacterial structure and how we can use that to tell different bacterial species apart. All right, so first and foremost, bacteria are prokaryotes. So like I said, that means they don't have a true nucleus. They have the nucleoid, which contains their DNA. There's no nuclear membrane or mitotic apparatus. The chromosome of a typical bacterial organism, like for example E. coli, is a single haploid double-stranded circular molecule of DNA, and that contains about 5 million base pairs. Replication, so I'm going to make this my single DNA. This is my attempt at making a supercoiled DNA double helix. Okay, so if this is my circular bacterial genome, all of the replication is going to start at a single point of origin, known as an origin of replication, or ORI. Replication is going to occur bidirectionally, right, in a semi-conservative manner. It's going to require a couple specialized enzymes. Topoisomerase 1 is basically going to unravel the DNA, and DNA gyrase is going to basically ravel it back up when it's done. So that's going to reintroduce the supercoils. And there are some antibiotics that we use that basically inhibit the activity of DNA gyrase. Nalidixic acid and quinolones, for example, bind to the bacterial gyrase, preventing DNA replication. And the bacterial RNA is transcribed by an RNA polymerase, just like us, and coupled with translation. The 5' end of the mRNA is being translated, while the 3' end is being transcribed. And there are drugs that get in the way of that too. For example, rifampin actually prevents RNA elongation. And basically it does that through the RNA polymerase binding, and that prevents its transcription. Bacteria have slightly different ribosomes than us as well. They have a 70S ribosome. The bacterial ribosome is actually a main target of antibiotics. 3 antibiotics in particular, chloramphenicol, lincomycin, and erythromycin, bind to the bacterial ribosomes and inhibit peptide bond formation. So basically, stopping that component. And then there's another class, the aminoglycosides, which includes antibiotics like streptomycin, kanamycin, and neomycin, which will bind to the 30S subunit, and that'll inhibit elongation. There are a lot of different structures that make up a bacterial organism. Some are shared by all of them, and some are only seen in certain organisms. The first that we'll talk about is the capsule. I'll go into a lot more detail about that, but all organisms have a capsule. The capsule is made of a polysaccharide, and it aids in adherence to tissues, but it also helps the organism resist phagocytosis. There's also something known as the glycocalyx, which sometimes we see instead of capsule. And again, this is going to help it adhere to foreign surfaces like, say, catheters. And it's made of polysaccharides as well. Now, in gram-negative organisms, which have a slightly different structure, you might see things like LPS, which is lipopolysaccharide, or O-polysaccharides, or porins. All of these have different functions, either to induce the immune response or transport bacterial products. I'll talk about those when I talk about the different types of organisms. There may also be a space called the periplasm that's found more in gram-negative organisms, because basically it's a space between the outer membrane and the cell membrane, and I'll explain what that means in a little bit. All of them have a cytoplasmic membrane. This is a phospholipid bilayer, just like this, and it's embedded with different binding proteins that are important to support the life of the organism. There are some structures that are used specifically for motility. So that could include like a flagellum, which is a proteinous structure that helps an organism move around. There's also pilus or fimbriae, which can be used almost like in a rowing fashion or used to mediate adherence to cell structures. And then also there's a particular type of pilus called the sex pili, which allows bacteria to exchange information during conjugation. And there's also something called spores, which I'll talk about at the very end. Spores are basically the escape pod for gram-positive organisms, and only two types, Clostridia and Bacillus. And they basically contain a complete copy of the bacterial DNA in case the bacteria needs it. So let's go through each of these in a little bit more detail. There is a table that lists all of these and their specialized structure function, as well as what they're made of in the course note. All right, so let's start with the bacterial cell wall. This is a thick, continuous barrier that surrounds all bacteria, with the exception of one, and that one is actually the organism Mycoplasma. Mycoplasma pneumoniae is a cause of kind of atypical pneumonia, but there are other forms of mycoplasma. All the mycoplasma do not have cell walls, but the rest of the organisms do. And the cell wall is actually what gives the bacterium its shape. And we can differentiate organisms kind of grossly based on the thickness of the cell wall. There are three main shapes that we tend to see organisms take. They can be spheres, rods, or spirals, which are known as cocci, bacilli, or spirochetes. It's not written out here, so I will write it. And you can see that there's variations on the theme. So we can see them as like diplococci or chains of cocci or clusters of cocci. We can see short rods, long rods. And then in spirals, we can see like corkscrews, or we can see like comma-shaped organisms like the vibrios. Hybrids of these exist. So for instance, Haemophilus influenza and C. trachomatis are very short rods that are sometimes referred to as coxobacilli. So it's kind of difficult to tell the difference between a coxo or a bacilli there. Sometimes we'll see like a rod, like Helicobacter, but it's got kind of this curved shape. So it's a spiralia. And this cell wall is really kind of playing a role here. The cell wall also interacts with the immune response and can be a target for antibodies. Bacterial cell wall can be used for serologic identification. So basically, whether or not a person has antibodies in their serum that is specific for that cell wall target. And that's what's known as serotyping, when you're seeing if the antibodies present are specific for a specific organism strain. So the cell wall has everything to do with the term gram staining. Gram staining is by far the most commonly used test in identifying bacterial organisms. It is so common that when a report is given, the report will be given something like this. Gram positive diplococci were found in the patient's sputum or something like that. And you'll see that they're telling you a couple things. They're telling you the shape. They're telling you that those organisms arrange themselves in pairs, and they're giving you the gram stain result. So it's a really important thing to understand. So first off, let's just talk about how you do a gram stain. Performing a gram stain is generally pretty quick and pretty easy. You're going to take your patient sample, you're going to heat fix it or otherwise dry it to a slide. You're then going to stain the slide with crystal violet, which is this really bright purple, beautiful stain that is precipitated with iodine. Then you're going to remove the unbound stain or the excess stain using acetone or an acetone-based decolorizer and water. So that's going to remove anything that isn't already fixed into the cell. You're then going to add a counter stain, safranin, and that's going to be used basically to stain any decolorized cells. And what you get is you get either purple cells, which are positive. So remember, P for purple is also for positive. Or you're going to get the cells that weren't stained by the crystal violet. They're going to show up red, just like the safranin counter stain, like this. And the reason that we see these differences in a gram-positive or a gram-negative organism has everything to do with the amount of peptidoglycan that is found in the bacteria's capsule. So gram-positive organisms have a very thick peptidoglycan structure. Gram-negative organisms have a very thin peptidoglycan structure, so they are not able to retain the crystal violet structure or the crystal violet stain. Now, this is not the only thing that is different between these two types of organisms. There are several structures that wind up being found more in gram-positive organisms or gram-negative organisms. So let's kind of take them one by one. I'm going to talk about peptidoglycan in the gram-positive organisms. There is some peptidoglycan in gram-negative, it's just much less. So within the gram-positive structure, cell wall structure, you not only have this thick peptidoglycan, but you have this lipoteichoic acid, sometimes referred to as LTA. It's basically a site of, it's basically a pro-inflammatory molecule within the organism. We only see it in gram-positive organisms. And basically, the LTA is what's going to induce an immune response. It's highly immunogenic. It's going to induce expression of things like IL-1 beta and TNF alpha. So this is part of what makes us respond to the bacteria in general. Let's spend a little bit more time, though, talking about the peptidoglycan, because as I mentioned, the peptidoglycan is kind of the biggest structure in a gram-positive organism's cell wall, and that's what's actually going to provide a lot of structure to it. So in gram-positive organisms, this peptidoglycan layer can be 20 to 24 layers deep. It's what retains that crystal dye, and it has this backbone of beta-1,4 acetylmuramyl N-acetylglucosamine or glycan, and that is cross-linked by peptide bond exchange, and that process is known as transpeptidation to tetrapeptides. So if you look over here, you have these tetrapeptides, right? And then you have a peptide cross bridge, and then you have either NAG or NAM. And they're basically forming this network to kind of create this woven fashion that provides the peptidoglycan structure. So peptidoglycan is basically a combination of beta-1,4 NAM plus tetrapeptides, or beta-1,4 NAG and tetrapeptides, all working together. This whole process of creating the peptidoglycan is catalyzed by membrane-bound transpeptidases and decarboxypeptidases. So these enzymes will basically remove unreacted terminal dealanines to limit the cross-linking. And the other reason this is important is that the transpeptidases and carboxypeptidases are called penicillin binding proteins. Now, you might not know what all of those things mean yet, but penicillin, surely you've heard of. And they are the target for penicillin as well as any other beta-lactam antibiotics. So later, when you're hearing Dr. Bradrick talk about penicillin binding and beta-lactam antibiotics, this is what they're actually targeting. This is where we're actually going to be doing work. And it's important because it tells you how important this cell wall is to the structure of the organism, that when we attack these penicillin binding proteins, we're actually able to harm the bacteria in the long run. The next kind of structures we'll talk about are gram-negative structures. These will be the pink or red stained organisms in a gram stain, as I'm showing here, and that's due to the safranin counter stain. Gram-negative bacteria do have a cell wall, but the peptidoglycan layer is much, much thinner, so the crystal violet stain is not retained. But it does have an outer membrane, which is a lipid bilayer, and then it has this periplasmic space, and it has a plasma membrane as well. The outer lipid bilayer basically excludes hydrophilic compounds and some antibiotics. And there's some lipid proteins in here. These lipoproteins are covalently linked to the peptidoglycan below, as are uh, other membrane proteins. Most notably that we need to think about is LPS, which just like LTA is a highly immunogenic structure, and LOS, or O antigens, and porins. And another term for LPS, I didn't mention it before, is endotoxin. So you might hear me refer to it either way. The periplasmic space is basically the space between the inner and outer membranes. It's going to contain hydrolytic enzymes, binding proteins for sugars, amino acids, and ions, and it might also contain beta-lactamase. And in some organisms, beta-lactamase is an enzyme that can actually destroy penicillin and will confirm some penicillin resistance on that organism. The last structure I'm going to talk about are spores. Spores, like I said, this is the escape pod for an organism. They're really only made by two genuses, Clostridia and Bacillus, and they're made when nutrients become limiting. So bacteria will undergo what's called vegetative growth, where they'll grow happily when nutrients are available. But as nutrients become limiting, the organism undergoes a process known as sporulation, which basically is it's creating this thing here. The spore can survive really adverse conditions. It's resistant to heat, to pH changes, a lot of chemicals. Um, when we talk about spores, when we talk about anthrax, I'll tell you the story of Lubia, the baby woolly mammoth that had B. anthracis spores on it that survived in, the tundra for 40,000 years. That's how hardy these structures are. But basically, it has this keratin spore coat, which is a protein. It then has this thick layer of cross-linked peptidoglycan. It has just normal peptidoglycan. It has a cytoplasmic membrane, and then in the core, it has the entire genetic material. So basically, this spore waits around until nutrients become rich again, and then it re-enters vegetative growth, creating the organism again. So it's a specialized structure that really makes these kind of hard to ever eradicate and also hard to kill. It's part of why like C. diff has been such an issue in hospitals, because C. diff is a Clostridia, so it produces spores. So you have a patient who has C. diff and they use the restroom, and you can try to clean it, but if there are any spores present, any of the antimicrobials that you normally use to clean that restroom will not be cleaned up. And then it can undergo vegetative growth if introduced into another person's gastrointestinal tract. So these are very cool structures, but for us, very bad news.